pharmacology here, who has a distinct privilege to have her passion revolve around something that I would suggest earnestly affects all of our lives in extremely meaningful ways. She studies the neuropharmacology of addiction and primarily focuses on the neuropharmacology of the addiction around alcohol. And I love in her abstract how there are two things in your abstract that stuck out to me. Just ultimately that the government condones the use of a chemical that is wildly addictive, and that there's a repertoire. Well, we'll tell you. We'll talk about it. I mean, we're not talking about nicotine. We're actually talking about alcohol, but we'll talk about. And that there's this repertoire of information around the neurotransmitters and the opportunity for understanding. And the statistic that jumped out at me of the alcohol use disorder, which is a term I don't think all of us are comfortable with and know, is really 7% of people are challenged. And I think to your last and final sentence in your abstract, there is great suffering in an addiction and a challenge being in a family member with any alcoholism or that conflict, that there's great hope for the kind of work you do. So I don't think we could have a better transition into your work and learning.
Thank you, Wendy. And I want to echo Alex and thank Larry for inviting me to this. This is a terrific event, and hope I will add to it or be worthy of it. Okay, so the title of my talk is Everyone Knows About Alcohol, but what do we really know? The everyone knows about alcohol, that part comes from my having done alcohol research for most of my career. So if somebody asks me what I do, I say I do alcohol research. And then usually or very often, somebody will launch into a story about, I have a family member who I think they drink too much, or maybe I drink too much, and I drank a lot last night. Is that going to hurt my brain? And on and on. So everybody is very familiar with alcohol. And yet, there are a lot of people who don't even consider alcohol to be a drug, much less an addictive drug. So there's quite a kind of a dichotomy about people using alcohol, knowing people who use alcohol, knowing people who have problems, and yet it's not really a drug because it's legal, you can buy it, it's not hard to get, so it's not like cocaine or anything like that. And yet it is an addictive drug. And the interesting thing about being in this session is alcohol is actually a physiological stressor. So when you drink alcohol, it activates the stress system in your body. And yet, we usually use it to relieve stress. And it does seem to relieve stress, so it's kind of a contradiction, but it's good for this session. What I had on the first slide is something that, again, not everybody is familiar with, and that is the equivalency of alcohol in every drink. So, for example, Alex goes home and drinks 3 glasses of champagne, which is like, whoops, like I turned my thing off. Which is like 3 glasses of wine. And you might think that if you drank a glass of whiskey, that might actually be equivalent to a couple of glasses of wine because of the volume. But actually, there's the same amount of alcohol in a shot of whiskey as there is in a glass of wine. And so sometimes, or if you drink beer, you kind of get full. You're not getting as much alcohol as you get out of a shot. So it's something that people need to be aware of, and they're not always aware of it. Okay, so in terms of statistics, this is from the National Institute on Alcohol Abuse and Alcoholism, NIAAA. They say that about 88% of adults have drunk alcohol at some time in their life. 71% drank in the last year, about 60% drank in the last month. 16.3 million adults, which is actually only 6.8% or 7% of the adults in the United States, have alcohol use disorder. Now, alcohol use disorder, that's just a definition, and it's changed over the years. So it used to be alcoholism, which everybody is pretty familiar with. Then it became alcohol dependence, and now it's alcohol use disorder. And I'm going to go through the definition of what that means in a minute. And interestingly, in terms of treatment, only about 10% of those people who need treatment get it. And partially that is because I don't think it's because of lack of insurance now, because the insurance does cover mental health and substance abuse. It's probably more, it has to do with attitudes. So many people and even physicians don't really think of addiction as a disease, or particularly alcohol addiction as a disease. They think of it as bad behavior, a moral lapse. Most people drink, most people don't become addicted, so what's wrong with you? Why can't you just stop drinking? It's easy to stop drinking. But when you're addicted, it's not easy to stop. And that's something that So there's still a stigma attached. And I think, and the other thing is that there's not really a very good treatment. So put those things together, people don't get treatment. Okay, so why do we drink in the first place? You heard a little bit about the reward system in the bees, but we also have a reward system in our brain. And you drink for mainly two reasons. Positive reinforcement. So reinforcement is something, well, something that makes you continue the behavior. So if you get a pleasurable sensation from drinking for whatever reason, because of the social environment that you're in, or because it just makes you feel good, that's going to make you continue to drink. The other reason is negative reinforcement. So that negative reinforcement means taking away something that's bad. So as I said, people will drink to relieve stress or to relieve anxiety. Alcohol is a drug that relieves anxiety. So if you come home and you're nervous and you're anxious, you're going to drink, and maybe you're just used to it. You'll take a drink because it's going to take away that bad feeling, and that's going to make you take another drink. So these are basically the two reasons why most people drink alcohol, and why I drink alcohol, actually. But most people, as I said, do not develop alcohol use disorder. This is the definition of alcohol use disorder. It's the current definition. It's a psychiatric definition. And so basically, it is telling you what the behaviors are that mean that you have alcohol dependence or alcohol addiction. You take alcohol in larger amounts or over a longer period than it's intended. You want to cut down, but you find that you can't cut down. You keep trying to cut down, but you don't. You spend a lot of time trying to get alcohol and to use alcohol and to recover from alcohol use. And then number 4 is one that just got put into this definition, but it's very important, and that is craving for alcohol. Now, it doesn't mean if you just happen to crave alcohol. Many people who drink socially say that I do have a craving for alcohol, okay? But it's different when you're addicted, and I'll talk about that later. And then a lot of other things that basically say that what's happening is you're using alcohol to the point where it's interfering with your life. You can't meet your obligations, you can't do your job, you can't take care of your family, but you still keep drinking, okay? And this is basically the hallmark of addiction. Now AUD is defined as, I knew I was going to do this. The little pointer thing is here. Okay, so it's defined as mild, moderate, or severe, depending on how many of these symptoms you have. So what this means is that the whole definition is very heterogeneous. So one person can have 3 symptoms, one person can have 2 symptoms, one can have 5, and they don't have to be the same symptoms. So what we are trying to study is what's the genetic basis for developing addiction. Well, that's really hard to do because not everybody is the same. And so when you take a whole bunch of people and they all have alcohol use disorder, they don't all have the same disease because they have different symptoms. So that makes it a little bit hard to study. Okay, so what do we know about it? What do we know about alcohol addiction? It's a neurobiological disorder, and this is where we get into trouble because people don't sometimes think that it has anything to do with the brain, but they just think it's kind of psychological. It's not really a change, but it is actually a change in biology in the brain. It's caused by excessive alcohol consumption, and the definition that I just gave you doesn't really say much about what causes it. It says what happens when you have alcohol addiction. These are the signs. So we know that both environment and genetics contribute to addiction. And certainly what Alex just talked about is pretty much wholly environmental things outside of you, the stresses in your life, all of those things, they are going to contribute to the occurrence of alcohol addiction. But the environment also includes availability of alcohol. If alcohol is not available, you're not going to drink, you're not going to become addicted. In some societies, alcohol use is frowned upon or it's not even allowed, and so addiction doesn't occur there. And then you have your individual stresses that contribute to it. There's about a 50 or 60% contribution of environment versus genetics. Okay, so genetics is 40 to 50%, environment 40 to 50%. But there is a genetic predisposition, and that's about 50%. What we don't know is what is the genetic predisposition to. So it could be that you have a genetic predisposition, something different in your genetics that allows you to or drives you to drink to excess. Or you may have something in your genetics that makes you susceptible to the changes that alcohol causes in your brain. And that's one of the things that we would like to figure out. And environment and genetics are going to interact. If you have a genetic predisposition to one of these things, then the environment's going to act on that as well. Okay, so what does alcohol do? Okay, so I said it's alcoholism or addiction or AUD is a neurobiological disease. So at the risk of completely oversimplifying, I just put up this slide to talk about neurons in the brain. So I have 2 neurons here, okay? The brain has 86 billion neurons. Okay, so I'm going to say what happens between these two, and then you can think about that happening everywhere. So you have, I got to really look at this. All right, so you have one neuron here. When it's activated, there's an electrical signal that comes down here to the synapse. At the synapse, you release a neurotransmitter, so that's what this is showing. So a neurotransmitter is getting released from this presynaptic neuron and acting at specific receptors on the postsynaptic neuron. And that's either going to enhance activity here or decrease it. And in addition to neurons in the brain, so you have 86 billion of those, you actually have more of these glial cells. And we talked about these a little bit before, but the ones I'm talking about are astrocytes and microglia, and then there are also other glia that form insulation on the neurons. The reason I'm talking about them is that they're very important actually for neurotransmission, okay? And so I think somebody mentioned that they're support cells, which is really what has been thought of in the past, but actually they're more active than that, and they play a pretty active role in the way the brain functions. So just to talk about the 2 major neurotransmitter systems, and the reason I'm doing this is because that's where alcohol works. The inhibitory system in the brain is mediated by the neurotransmitter GABA, which stands for gamma-aminobutyric acid. And basically, GABA is released from a presynaptic neuron. It acts on the receptor on the postsynaptic neuron. And these receptors these are different protein subunits that make up the receptor. And depending on the composition of these receptors, they are sensitive to alcohol, or to things like benzodiazepines or anesthetics. And then the glia play an important role because they basically recycle the GABA back into the presynaptic neurons. So the glia are actually enhancing the function. And GABA, again, is the inhibitory system, so when it acts on this neuron, it inhibits its activity. The other major neurotransmitter system in the brain is the excitatory one, and that's glutamate. And this is basically the same kind of picture. So glutamate is produced in the presynaptic neuron. It interacts with receptors on the postsynaptic neuron. And the one I'm talking about is this one, the NMDA receptor, which is basically shown here. It's also made up of different subunits. And the importance of the subunits is that, well, there's a lot of importance, but in terms of alcohol, depending on the subunit composition, the receptor will be more or less sensitive to alcohol. So alcohol, the reason it's difficult, one of the reasons it's difficult for people to study is that it is not like other drugs which act at receptors. So, for example, cocaine. Cocaine acts at a specific protein in the brain, okay? So it has a site of action. It binds to that protein. So you know where cocaine's going to go and what it's going to do. Alcohol is not like that. These systems are sensitive to alcohol, but they're not specific for alcohol. So alcohol acts on a lot of different systems in the brain, but these are the most sensitive to it. And the glutamate system is like the GABA system in that the glial cells are also important for recycling the glutamate and bringing it back into the neuron. Okay. So what does alcohol do on these systems? So this is a picture of the effects of alcohol acutely, okay? So when alcohol is present in the brain, okay, in terms of the inhibitory system, the GABA system, it enhances the ability of GABA to act at these receptors. And it depends, again, on the subunit composition. Some of them are more sensitive to alcohol. But basically what alcohol is doing is increasing inhibition of neuronal activity, okay, at the GABA system, at the GABA neurons. On the other hand, on the glutamate neurons, alcohol actually inhibits the ability of glutamate to interact with these receptors. So it inhibits the excitatory system. So when you have alcohol here, you're getting more inhibition. And when you have alcohol here, you're getting less inhibition. So overall, you're getting an increase in inhibition, a decrease in excitation. So alcohol is a depressant, even though it might not feel like it when you drink it, but it is a depressant. But because you have 86 billion neurons in your brain and they act in circuits, what alcohol does in any particular brain region depends on how these neurons are interacting with each other. So it's going to change the balance of inhibition and excitation. And that can lead to, for example, we talked about this morning about dopamine being important for mediating reinforcement. Well, alcohol increases dopamine release just like the other drugs do, but it does it mainly through acting through these systems. Okay, so that's what happens when you take a drink, okay? Or 2 drinks or 3 drinks. Okay, but if you drink chronically, or if you're given alcohol chronically if you're an animal, and you have it on board for a long time, then changes occur in the brain, okay? And these changes are basically compensatory changes. So normally, when you have alcohol in there, it's enhancing this response. So the brain adapts to that, and it reduces the GABA system. It reduces the activity of the GABA system. Okay, so when alcohol is there, it's enhancing this, and it's kind of bringing it back to normal. So that, you know, you have alcohol there for a long time, the brain reduces that activity. If you keep alcohol there, it'll potentiate it and keep it normal. When you take alcohol away, though, you have less inhibition. So you end up with hyperexcitability, anxiety. On the glutamate system, it's the other way. So alcohol acutely inhibits this system. When you have alcohol there all the time, then the system adapts and it upregulates. So there's more activity here, but alcohol's inhibiting it, and that keeps you in the normal range. But when you take that away, you get excessive activity. So here you have less inhibition, and here you have more excitation. So, and actually, just as a side thing, the changes, these look like changes in the numbers of receptors, which probably happen, but a lot of it has to do with changes in the composition of those receptors and how sensitive they become to alcohol. Okay, so when this happens, these changes occur after you have prolonged exposure to alcohol, and they contribute to withdrawal signs. So 2 of the things that I didn't mention in the definition of AUD are the development of tolerance and withdrawal when you take alcohol away. And this can happen after a relatively short time or after a long time, but these particular changes are reversible. Okay, so you can have withdrawal signs, maybe mild withdrawal signs, but these will reverse if you don't drink alcohol. But when you drink alcohol for a longer and longer and longer periods of time, these are the early signs, but the changes become more and more excessive, and they become more permanent. Okay, and these are the beginnings of the changes that lead to alcohol addiction or alcohol use disorder. Okay, so the thing is that we know that this happens. We can see it in animal models, and even if we look at human brains after death, we can see some of these changes or similar changes. So we know that that happens after heavy and prolonged alcohol drinking. And the changes that I've just talked about are probably involved in the transition to becoming addicted. But that doesn't tell us whether you have a predisposition to that or not. So it happens. It may happen to everybody, or some people may be more susceptible to it. The other thing that we know, and this is again a duh thing, if you drink more, you're more likely to become addicted to alcohol, okay? You could guess that, but there's actually data showing that. So if you, this might go back. Okay, If you get up to drinking more than 5 drinks a day, you have about a 60% chance of developing alcohol addiction or alcohol use disorder. And it's interesting that there's nothing in the definition of alcohol dependence or alcohol addiction that talks about how much you drink. It says that you spend more time drinking, that you can't cut down on drinking, but it doesn't talk about the fact that you're drinking a lot or you're drinking a little. That's just not in there. But it's clear. Those on the bottom, this is average daily volume. It's actually drinks, average drinks per day. Drink number, yeah. Okay, and this is, yeah. So when you get high, this is 5 drinks, 5 or more drinks, which is a lot per day. You're more likely to become dependent. Okay, so this, again, it doesn't tell you whether there's a genetic predisposition to this or not. So we know 2 things. We know that these changes occur in your brain, and we know that if you drink more, they're probably more likely to occur, but we don't know how much is environmental or what is genetic. So we decided to do a pretty simple study, and this is using an animal model, which is called, I'm just not good at this. Okay, called the alcohol deprivation effect. So we are measuring in this study 2 different behaviors or 2 different phenotypes. We're measuring in an animal model voluntary alcohol drinking, and then we're measuring something that is called abstinence-induced alcohol drinking. So in this cartoon, the rat is in his home cage, and we actually do have enrichment for the animals, but they're not television sets, but they have something that they can do. So the rat is in his home cage, and he has a choice of either drinking alcohol or water, that we give to him. So, he'll drink alcohol when he feels like it, and he'll drink water when he feels like it. And with these rats, this can go on for days, for weeks, for months, or years, depending on how you want to do the experiment. But they will actually get to the point where they drink a certain amount of alcohol, and it's pretty steady. So it's just, every day they drink a certain amount of alcohol, and we can measure that. But then one day you take the alcohol away. Okay, so the rat notices that he doesn't have alcohol, and he's a little bit upset about it. But there's nothing you can do about it because he's in a cage and you have to give it to him. And then at some point you put the alcohol back. Okay, so now this rat, who before had been drinking at a steady level, he just chose a certain amount of alcohol or he chose a certain amount of water. When he gets the alcohol back, he just drinks a huge amount until he's knocked out, okay? And that's supposed to be a model of alcohol craving. So if you stop drinking, you've been drinking, you stop drinking, and then when you have the opportunity to drink again, you have this strong craving to drink, and you drink more than you normally would if you're a rat or even if you're a person. And so the good thing about this is that it's quantitative. So we can measure how much they drink voluntarily, and then we can measure how much they drink when they have this alcohol deprivation effect. And that's useful for someone who wants to study a genetic characteristic, because you have to have something quantitative to measure. Okay, so what we're trying to determine is, are these phenotypes heritable? Is there a genetic component to these phenotypes? So to do that, we have to use more than one strain of rats. So the strains of rats that we use generally, or mice, are inbred. And that means that they are all genetically identical, okay? So you could use a whole bunch of inbred animals, but what we use is something that's called recombinant inbred rat strains. And they have, each strain, all the animals are genetically identical, but then there's a whole panel of strains that have a different genetic makeup. And we look to see, does the phenotype vary across these strains? And if it does, we can determine whether it's heritable. So the good thing about using animal models, the good thing about it is that we have the opportunity as experimenters to hold the external environment constant, as constant as we can. So they're all kind of living under the same circumstances, although internal environment can vary, but we can't do much about that. So what we can do is we can measure the phenotypes in these animals. And so if you say you've got 6 rats in a strain, you can, if there's a genetic component, all those rats are genetically identical. The amount of variability among those rats in that strain is going to be less than the variability across strains. So there's going to be more difference. All of those animals in the one strain are going to be the same, but another strain might be higher or higher. So you can tell if there's a genetic influence on the behaviors. And when we did this, what we found was that the amount of alcohol that they drink when they have the free choice to drink, that really was highly heritable. So that showed about 50% heritability, which is about the same as heritability for a lot of neuropsychiatric diseases. So that was actually where we saw the genetic characteristics. The alcohol deprivation effect, where they drank a lot when the alcohol was given back to them, that was not very heritable. Okay, so that, what you would say, has more of an environmental component, whereas just the amount of alcohol that you drink voluntarily has more of a genetic component. Okay, so how do you determine? So you say, okay, well, it's genetic. So what? So how are we going to determine what the genes are that are important for this? And that becomes difficult, but somewhat doable. So how do we find genes for any trait that we're looking at? Well, you know that now we can sequence the genome, so we can actually measure every base in the genome. But even before that, you could determine variants, genetic variants, and you could find them in a human population, you can find them in an animal population, and you can determine which of those genetic variants are associated with the disease that you're looking at, okay? And you can do it dichotomously, so either you have the disease or you don't have the disease and which ones are associated, or you can use a quantitative trait like drinking, and you can find out which of those variants basically correlate with the amount that the animal drinks or that the human drinks. So that's good. So you can find what that tells you is, what it gives you is the parts of the genome that are important for the behavior that you're measuring. So there's some region in the genome. It doesn't tell you the exact genes, though. So how do we get a little bit closer to that? So the dogma, again, was that DNA makes RNA, messenger RNA, that makes protein. So the idea would be that, gee, that would be good if we find a variant in DNA, it'll change the function of this protein, and then we'll know what's important. But as it turns out, only about 1.5% of the genome codes for protein. So when you look for these variants, most of them have nothing to do with proteins. They're in regions of the genome that don't make proteins. But the genome, the DNA makes all kinds of other RNAs. So it makes messenger RNA, but it also makes RNA that's part of the promoter region for the gene or part of another region of the gene that regulates the gene's expression. And it makes non-coding RNAs, which also regulate gene expression. So when I say it regulates gene expression, I'm talking about the messenger RNA. Messenger RNAs vary in levels, okay, across people and across rat strains. So one rat might have a low level of messenger RNA for a particular gene, another rat has a high level. And that, of course, can be regulated by the environment, how much mRNA you make, but we try to hold the environment constant. So we can find out, we can measure these messenger RNAs and do the same thing we did with the behavior, and we find out that they're actually heritable. So the level of messenger RNA that's produced, the transcriptome, which you heard about before, that is genetically controlled to a great extent. So what we can do is we can find the variants that actually control the levels of the transcripts that we're interested in. And if the same region of the genome controls the behavior and also controls the level of the RNA, then we're going to say, okay, that's a candidate for the behavior that we're interested in. So that's how we can get not only to the region of the genome that's important, but also to, I don't know when I started, also to some idea of what the genes are that regulate the behavior. Okay, so now I'll just show you what we do. And there's one important thing that I need to mention. Okay, so this is the data that we use to do the heritability. Okay, so these are the different strains of rats, and this is how much they drink. They drink different amounts. And that's how we determined that there was about 40% heritability for drinking. And then we can do this analysis, which is called quantitative trait locus analysis. It's similar to a human GWAS. You find the regions of the genome-wide association study. You find regions of the genome that regulate the behavior, okay? So we did this, and we find these regions of the genome. But one of the important things is that we're not interested in what alcohol does to the transcriptome. We're interested in what's the baseline difference between these rats that predisposes them to drink different amounts. So if we used 6 rats per strain to do this experiment, we can take another 6 rats from this strain that are genetically identical, but they've never been exposed to alcohol. So we can then look at them and try to find what are the genes in these rats that lead them to drink when they're given alcohol. Okay? So basically, this is what we do. So we take all these different strains of rats, a bunch of them per strain, and we measure gene expression levels, RNA levels, in their brains. They haven't been exposed to alcohol, and because we're trying to find the genes that affect the predisposition. And you already heard about gene clustering. We know that it's not one gene that controls a complex trait like drinking or like dependence. It's a lot of genes. One way to look at this is to cluster genes according to their expression levels across these different strains of rats. And when you do that, you get gene modules that turn out to be, and it's interesting, you might not think it, but they turn out to be indicative of functional pathways. So you get these modules of genes by doing this clustering. And then the important thing here is, say you were looking at one gene and you thought that the expression level of that gene is important for the behavior. Well, you'd like to say, okay, if that's true, then the expression level, the mRNA level for that gene is going to vary, is going to correlate with the behavior. So, for example, if a rat drinks only a little bit, the expression level of that gene will be low. If it drinks more, the expression level of that gene will be higher. So you try to find genes, only instead of doing individual genes, we do these clusters of genes that correlate with behavior, okay? And so that's the second step. So we find the genes, the modules that cluster and correlate with the behavior. And then we do the same thing that we did for the behavior. We determine the regions of the genome that regulate the expression levels of these genes. And we look for overlap, which is what you're seeing here. And by doing this, so we're really looking at which DNA variants, which regions of the genome control drinking, and the same regions that control drinking control the expression levels of these genes, okay? And that gives us candidate genes that are important for drinking behavior. Okay, so this is the module that we ended up getting, and doesn't mean anything. It's a bunch of genes. One of the interesting things is that this gene is very much correlated with or connected to all of the other genes, and it's something that nothing is known about. So we don't know anything about its function. When it says no annotation, it just means nothing is known. We do know a little bit. We've looked at its sequence and its structure, so I can tell you that it seems to be a non-coding RNA. It's not one that makes a protein. But we do know about the function of a lot of these other genes that are named. And so when we look at what are the functions of the genes in this module, we came up with some interesting things. A lot of these genes are found in glial cells, not in neurons. And if you look at their overall pathway functions, you find that actually the genes are involved in the function of the GABA system and in the function of the glutamate system, which is really pretty interesting. So this is a module. The genes are involved in glial neuronal communication. And the rats that are predisposed to drink high levels of alcohol at baseline have lower function and lower GABA function and higher glutamate function, which kind of makes sense because when they drink, that's going to be somewhat corrected, okay? The other function that we found for these genes was really also very interesting because it has to do with inflammation and the immune response. So the microglia are the immune cells of the brain, and a lot of these genes are found in these cells. And so this has become a very highly studied area recently for neuropsychiatric disease, and for alcoholism, or for alcohol drinking. The fact that neuroinflammation, inflammation in the brain, is believed to be related to various neuropsychiatric diseases. And in our case, we came to it through an unbiased way, but we found that all of these genes, or many of these genes, are involved in the immune response. So we don't know, does that mean that you have a different immune response and therefore you drink, or maybe drinking enhances the immune response, or it causes inflammation, and that's what stops you from drinking? And we don't really know that, but that's something that people are studying. So because the genes that we found are involved, they're supposed to predispose the animal to drink. So we wanted to check that, see if we could determine if that was really the case. So that gene that I mentioned, which is the most highly connected gene in this module, if it was expressed at a high level, that was an animal that didn't drink much. So we knocked that gene out in a rat. And so the wild type is expressed, it drinks this much. When we knock it out, it actually increases their drinking. So this is, pretty interesting in terms of targeting genes that might be important for alcohol drinking. Unfortunately, we don't know particularly what this gene does, but that's something that we have to study. It actually doesn't have any annotation in any, it's found in humans, and it's found in mice and rats, but nobody knows right yet what it does. But the question is, does any of this have anything to do with humans? Because they're rats. Well, it turns out that alcohol consumption is also heritable in humans, so it's a heritable trait. And if you look at studies that have been done from genome-wide association studies where they find the regions of the genome in the human that are associated with alcohol drinking, at least 2 of the things that have been found are what's called syntenic with the rats. So this is human chromosome 11 has basically the same genes that human chromosome 1 does, that rat chromosome 1 does. So they find basically 2 areas that we found in the rat, which are also come out in the human for drinking. So this gives us an idea that some of the genes that we have found, because the same genes are in the same region of the chromosome, some of the genes that we found in the rat may also be important for human drinking. Okay, so what I am saying from this work that we and others have done is that the predisposition, the genetic predisposition, has to do with the amount of alcohol that you drink. Okay, once you drink that alcohol, you're going to have these changes occur in the brain, and for some people, that's going to lead to development of alcohol addiction. If we can understand the genetic factors that predispose to drinking, it's possible that we can then have a target to reduce drinking, and that should reduce the incidence of alcohol addiction. But on the other, there's a couple of caveats about this. First of all, I can't run this thing. First of all, not everybody who drinks heavily develops alcohol use disorder. So you saw that in that graph, 5 or more drinks a day, you have a 60% chance. But that means you also have a 40% chance of not becoming addicted. So it's, as they always say, genetics is not destiny. So there are other things that are involved besides these genes that we're finding. The other thing that when we did this study with the rats, we wanted to look at craving, because craving is a really important factor in relapse drinking. So we chose a model of craving. But there are lots of other symptoms in the alcohol use disorder definition. Tolerance and dependence, tolerance and withdrawal, for example, and we know from our own studies and other people's studies that tolerance does have a genetic component. So it's not all as straightforward as it may seem from this, but the fact is that craving, which is really, like I said, a very important part of relapse, didn't seem to have a very high genetic component, but we know that craving, particularly in dependent individuals, is triggered by environmental factors. All of the factors that Alex talked about, those are the things that make people go back to drinking once they have become addicted, and even cause craving in people who are social drinkers. So these are obvious. It's obvious that it's 50-50. We can find genetic markers and genetic predisposition to certain aspects, but environment also plays an important role, and they interact with each other. And finally, I will. So the conclusion is that, I probably could have started out by saying both genetics and environment are important, but if we can find the genetics, that'll help a lot for a lot of people. Okay, and so I want to acknowledge my colleagues, some of whom are sitting in the audience, and also our funding, which is mostly from NIAAA. That's it.
That was great. One thing, I work in the media, and I think a lot, we're always looking for these quick tips. So, you're talking a little bit about the relevance of genetics and environment. And what tips do you have for people that know they have the genetics?
Well, if they knew they have the best thing, if you're really worried about it, would be not to drink.
Yeah, like complete abstinence, you mean?
Well, that would be one way to not get addicted, yes. I mean,
It would. Maybe until the environment and the stress took hold.
Because, well,
Yeah.
I mean, if you were really predisposed the way that shows, with the rats, I mean,
Yeah.
to drink a high amount of alcohol, the chances are that once you start drinking, you're going to drink a lot.
Yeah.
And if you drink a lot all the time, then you have a very good chance of developing addiction. So,
Yeah.
I think that would be, but there's really no way right now to know whether you are genetically predisposed or not, just because your
Even
parents,
if you're looking around at your family.
yeah, because there's still an environmental component.
Yeah.
So.
Yeah. Okay.
So just a quick question about the environment that the rats were in. So were they isolated or were they like?
They're isolated.
Okay, got it.
Because we have to measure how much
Yeah.
they drink. So, yeah.
Got it.
And that's another thing. That's a stress.
So is there
just-
an issue? Can you not measure how much they drink individually in a group environment as well? Could you not measure how much they drink individually in a group environment as well?
It's really hard to do because you only have the one bottle, and so you don't know who drank what.
You have
so,
like RFID on each rat.
So it's what?
You have like RFID on each rat and like measure the presence of the rat near the bottle.
Well, you could, yeah. I
the
mean,
depletion.
you could. The only thing you could do would be to measure their blood alcohol level if they drank enough. So that would be one way if they were all drinking, in a group. If you could get them at the right time and get how much you would know what the blood alcohol levels were. That would be one way to do that.
Got it.
But otherwise, it's really hard.
We're going to do a couple more, and then we'll have a break. Do you want to pop up there, and I'll go over here.
Thank you so much for the science. So much of the problem around addiction is that we're in denial, and across demographics, we don't tell the stories, and we don't talk about it. I was wondering, as a scientist, if you could comment on those relationships between the science of addiction and our unwillingness to talk about these issues.
That's really a good point. I mean, denial is one of the major factors, and there is a stigma to addiction, no matter what you say. And so people are really, even with a doctor. I mean, one of the things in terms of, you go to your doctor, you've got a little thing to fill out. How many drinks do you have? Well, people will say. So somebody will say, I drink 5 drinks a week. They really mean they drink 5 drinks a day, but they don't want to put it down. They don't want to have it in writing. They don't want to tell the doctor. And this is a problem, and it has to be made more open, I think. People have to be, if people start to think of it as a disease, a chronic disease, then a lot of the stigma is gone.
But even our doctors are not trained to talk about these things.
I think that's true, and we have courses, and we do talk to them, and psychiatrists are trained to do it, but they don't. A lot of times they won't push. You're asked a question, you give an answer. The doctor doesn't say, oh, "Come on, you don't drink," So, and that I think, but the more we talk about it, and the more we get it into the papers, and the more it's out there, it becomes less stigmatized, I think.
Thank you, Paula, Dr. Hoffman. My great-grandfather was a Quaker preacher, so he didn't drink. But then, interestingly enough, my grandfather was a First World War veteran, and he was a drinker, and there's actually an advertisement of him drinking. And so it kind of seems like military and alcohol, they kind of go together. My father was a Korean War veteran, and so on and so forth. But my question is, as far as the genetics of it, or the genome of it, it's kind of, I guess my question is, trying to get to the gist of it, is the diseases of society. Like a lot of our conditions are brought about by environmental factors, toxins, so on and so forth, that we're becoming more aware of. So with alcoholism, I guess you can have a predisposition for it, but is it really a kind of a disease of society? In other words, if you go back far enough and you could do analysis of people before alcohol was brought into society, but it goes back thousands of years, obviously, so thank you.
So the question is it genetic, or is it really something in
Well,
society?
is it a condition that we've kind of brought onto ourselves? Is it a disease of society rather than
You know,
some sort of abnormal birth abnormality or something?
well, I would say that, again, we're talking 50-50, so and the society plays an important role in that. One of the things I always remembered from my organic chemistry book was that they said people learned how to make alcohol before they learned how to make soap, okay? So it's obviously in societies where it's allowed, it's been here for a long time. Yes, I mean, it's both. But if you have that predisposition and you're in a society where alcohol is legal, like it is here, then it's available to you, and then you have the higher risk for that. It wouldn't hurt if you don't want it to happen to you.
I think we have one more over here.
I just wanted to make a comment and ask a question. The comment is this. One of the big differences between this condition and other psychiatric disorders, all of which are horribly stigmatized, by the way. So schizophrenia is a horribly stigmatized illness. It's really a disease. Everybody acknowledges it's a disease, but nobody wants to admit that it runs in their family or that they have it. But going back to alcohol, one of the differences is that patients who drink alcohol like it. Patients with schizophrenia don't like it. People with diabetes.
Well, I would argue with that a little bit, because it may be that when they're drinking at the beginning, they do like it.
Yeah.
When they're addicted to it, they don't like it.
Well,
the
reason they lie to you is they want to continue. Now, I agree with you that it's an effect of the alcohol that makes them want to continue. They would like to, each morning they wake up and say, I'd like to give up drinking. But they crave it. And, we for a long time thought that the reason people use drugs is because to avoid the withdrawal. But actually, people get off drugs for a while and they go back to them because they miss the effect. And that's why patients aren't honest with us. I push patients very hard on their drinking, but they're deceptive partly because they don't want to give up the behavior. They want to give up the consequences. I'm not, by the way, I'm not, and one of the things that this kind of genetic work does that's really useful is it does destigmatize this and makes it more likely that people can come in and say, this is a thing I have to fight because I have this disease. So the question is, how much of the genetics in animals that you're exploring in rats do you think translates to humans, given the complexity of the human behavior of drinking alcohol?
In some ways, I think it does translate, because it has to do with the reward system, right? And what happens to the reward system, whether you go from, one theory is you go from positive reinforcement initially to negative reinforcement later. And I think you can see a lot of that same thing in animal models. I mean, they're not perfect, and they certainly don't recapitulate the definition of alcohol use disorder. I mean, a rat doesn't really have obligations that it's not doing. But in certain aspects, I think in the genetics of it, I think there's quite a lot of similarity. So hopefully, I mean, I actually believe that the targets that we find, at least some of them, or the pathways, maybe not the specific gene, but the pathways that we find, inflammation, for example, in animals are going to be also found in humans.
Let's give Dr. Hoffman great thanks. And we'll break for about a half hour, but Larry Gold and his team have asked us to be back a bit before 4 o'clock so we can start right on time. So that gives us about 20, 25 minutes.